I was hoping to hear your voices of excitement. We meet on a very special occasion. My name is Mosamo Shabela. I'm the Vice Chancellor of UCT. And we have the pleasure of coming together this evening to celebrate. It's always a great pleasure when we have to host one of these evenings with a UCT inaugural lecture. And this evening, we are coming together to celebrate an important academic milestone in Professor Res Altwegg. Everybody knows him as Res. Now, Res to me invokes residence, so I wouldn't. Res, we are here to celebrate your career, and we want to take this opportunity this evening to honor your spirit of inquiry, your efforts in collaborative work that you've done over the years, and the impact that you have made that also contributes to the definition of excellence that is UCT. I want to take this opportunity to welcome all of you that are here today, to welcome the friends and family and colleagues of Res and all the invited guests that are here. I want to welcome our staff, our students that have joined us. And thank you for being here because you all make this evening what it is. Your presence here reflects the community that has supported the journey that Res has traveled over the years, and also symbolizes the journey that many of our established, distinguished academics travel. Let me take this opportunity to acknowledge the platform party. You may be aware of who our Deputy Vice Chancellors are, but if you are not, I will introduce you to Deputy Vice Chancellor Elelwane Ramukondo, who leads our Student Affairs, Transformation and Social Responsiveness portfolio that we are soon going to hopefully refer to as People, Culture and Society. We don't know. Our recently installed Deputy Vice Chancellor for Teaching and Learning, Professor Brandon Collier-Reid, who I can assure you that he has hit the ground running. And you know that when SVC, I can start sending him out to represent me in various meetings and committees, especially outside of the university and in national meetings. He recently came back from defending our enrollment targets with the ministry. So thank you for that. Our Dean of the Faculty of Science, Hussein Solomon, that I'm sure you all know. Our inaugural lecturer will be introduced formally shortly, but let me also introduce you to Associate Professor Freedom Gumedze, who's the head of the Department of Statistical Sciences, and he will offer the vote of thanks. Ladies and gentlemen, inaugural lectures at this university represent a time-honored tradition. They serve to confer the title of full professorship, even though Res was promoted 2 years ago, but we cannot miss this moment to formally celebrate your academic distinction that you have earned through the years and through dedicated research, teaching, and service. They provide all of us as a community with an opportunity to learn from and engage with the research and the work of our inaugural professor. Tonight, we'll delve into a field that sits at a fascinating crossroads of disciplines. And I like transdisciplinary work, just so you know. And in his lecture titled Adventures in Statistical Ecology, Professor Altwegg will take us on an intellectual journey through a domain where data meets environment, where models will meet the wild, and where science meets some of the most urgent conservation questions of our time. Professor Altwegg personifies the kind of academic leader and innovator that we are proud to celebrate at UCT. He's contributed tremendously to this university scholarly and is the founding director of the Center for Statistics in Ecology, Environment, and Conservation. And he continues to shape how we understand and respond to ecological change, biodiversity loss, and environmental sustainability challenges. In a world increasingly shaped by climate uncertainty and environmental strain, the role of interdisciplinary science, grounded in rigorous data and collaborative inquiry, has never been more critical. Just yesterday, we were celebrating similar interdisciplinarity of institutes at the Faculty of Health Sciences. So, Professor Altwegg's work reminds us that solutions to our most complex challenges in the world today will not be found in our silos, found in isolation, but in the fertile ground where disciplines meet and where diverse perspectives converge. Tonight's lecture promises to enlighten and provoke and to inspire and inform, and we are very much looking forward to it. He's a little bit nervous, not too much. And I encourage all of us to listen and listen closely, to reflect and think deeply, and engage meaningfully with the ideas presented afterwards. Without further ado, let me invite Professor Hussein, who has the pleasure of introducing our inaugural lecturer. Thank you. Round of applause.
At this point, we adjust the mic for somebody who isn't as tall. Good evening to all of you. Thank you. Okay. Thank you. So good evening. And firstly, I want to thank everyone for coming to this event. I think it's a very important event for the faculty and the university. And I have the honor at this event of introducing our speaker for tonight, Res. And I should say, taking a leaf from the Vice Chancellor, as I pondered on this, Res does not invoke residence in my mind, but it invokes research. Because as I was editing my notes, I constantly thought about research as I'm typing in Res's name, which is quite appropriate for what we are here for tonight. So Res Altwegg is a professor in the Department of Statistical Sciences in the Faculty of Science at UCT. He is, as you have heard, the founding director of the Center for Statistics in Ecology, Environment, and Conservation. I have to look at my notes. I know it as SEEC, which Res has led for the last 11 years. Res's background is in ecology. He studied biology at the University of Basel in Switzerland, and he did his diploma under the supervision of Professor Steve Stearns and Professor Bernd Erik Steder at the Norwegian University of Science and Technology. His PhD thesis was in evolutionary ecology, supervised by Professor Uli Reyer at the University of Zurich. He experimentally examined how perceived predation risk affected life history decisions in frogs. And I have to say that's the first time I learned that frogs make life history decisions. Res then pursued postdoctoral research under the mentorship of Professor Brad Anhold at the University of Victoria in Canada. He worked on a microbial system that allowed him to examine the effect of inducible defenses on population dynamics experimentally. Over time, he came to realize the power of new statistical methods that allow us to estimate key ecological quantities. And having gone from Europe to North America, he finally saw the light when his next postdoc was in the Department of Statistical Sciences at the University of Cape Town, where he worked on demography and species range dynamics in birds under the mentorship of Professor Les Underhill. His first permanent position was as the specialist scientist at the South African National Biodiversity Institute, which we know as SANBI, where he built up a research group in statistical ecology, working on various projects in climate change and applied biodiversity research. Since 2013, though, he has been at the Department of Statistical Sciences at UCT, first as an associate professor and then as a full professor from 2023. He teaches first and second year statistics courses for science students. And I can confirm that science students will tell you these are difficult courses. Res has also been involved with a third year course in quantitative biology for the Department of Biological Sciences. And you'll hear a bit about this as well in the other comments that we have to make. Res has also held a research chair with ACDI, the African Climate and Development Initiative, from 2014 to 2016. Very crucially, he led the establishment of SEEC, and he currently co-leads the group. This center brings together ecologists and statisticians with the aim to address some of the most important questions in ecology and conservation using cutting-edge statistical methods. SEEC, incidentally, has recently been re-accredited as a research group. In that re-accreditation, the team found that their work was without question of a very high quality and actually recommended that they continue and maybe even expand on their activities, which is a very positive indicator for the work that they are doing. They currently have 29 scientists, some as core team members and some as affiliates, and around 30 postgraduate students. The SEEC group is considered to be one of the largest research groups in this area around the world and the only one in Africa. Res has mentored 23 postdocs, supervised 17 PhD students and 24 master students. 26 of these have already graduated. In terms of research interests, I know that Res is going to speak a lot to this, but his interests span the broad field of statistical ecology, particularly population ecology, wildlife demography, and modeling species distributions. Over the last years, his research has focused on understanding the effects of climate change and other pressures on the population ecology and range dynamics of various organisms. His research has led to 151 peer-reviewed publications so far. And according to Google Scholar, 2 hours ago, he had the current stuff. He has 7,988 citations with an H index of 51. Now, I've looked at the trajectory. I think next week he'll cross the 8,000 line. He currently holds a B2 rating from NRF. So working at this intersection between ecology and statistics, his research is inherently interdisciplinary and collaborative. He has a strong network of international and national collaborators. He serves on various editorial boards, notably on the editorial board of Methods in Ecology and Evolution as an associate editor, and the editorial board of Ecology as a subject matter editor for the Statistical Innovation section. Previously, he was an editor and a handling editor, respectively, for the journals Animal Conservation and Population Ecology. He's considered to be a leader in this field. He was instrumental in winning the bid for SEEC to host the International Statistical Ecology Conference in Cape Town. This conference, ISEC, is the largest gathering in the field worldwide. and it attracted 500 delegates to the event in Cape Town in 2022. Being selected to host this event was a milestone for SEEC and for Res. This is held every 2 years, and this was the first time the event was held on the African continent. Res chaired the local organizing committee, but also chaired the scientific planning committee for the conference. Finally, I want to say that the distance from PD Han, where Res's SEEC group is based, to the John Day building, where the biological sciences are based, is about 10 meters. In my opinion, the intellectual distance is almost infinite. But what Res has shown us is that we can bridge the gap between these disciplines. And in this world that we would otherwise think of as being a collection of silos of different disciplines. In bridging that gap, Res has also showed us that we can do it and establish excellence in research. He also contributes regularly to the faculty and the life of the university. And in my opinion, among his peers, he has the position of quiet and unassuming leadership. So for all that Res has given to UCT, for all the work that he has done, we have gathered here today to listen to him and to honor the contributions of Professor Res Altwegg. Congratulations, Res.
Yeah. Thank you, Vice Chancellor. Thank you, Dean, for these nice introductions. Does this sound right? Yeah, can you? Okay, good. Screen, good. Okay, so the famous statistician John Tuckey once said, the good thing about being a statistician is that you get to play in everybody's backyard. And so I'm going to show you around my playground tonight. So if the topics feel a little bit disconnected, that's why. So first, we need to ask, why should we even worry about ecology? Why should we worry about the environment and conservation? And here's one answer from the World Economic Forum. So they give out a global risk report every year. It's not quite in focus. Is it okay? Yeah. Okay. So their global risks report, they ask people, economists. So these are the leaders of our economy and politicians, and they ask them about the top risks to the global economy over the next 2 and the next 10 years. And if you look at this on the right-hand side, the top 4 risks are all environmental. So you might not be able to read this. This is extreme weather events, and the second is biodiversity loss and ecosystem collapse. The third one is critical change to Earth systems, and the fourth one, natural resource shortages. So these are not people who are necessarily worried about nature per se. They worry about the global economy, but they understand that our livelihoods very much critically depend on our environment. And so if they are worried about it, we should be worried too. So the other thing is that all of this plays out against the backdrop of a worsening climate crisis. And the Intergovernmental Panel on Climate Change, IPCC, uses what they call shared social economic pathways to build their scenarios on, where they can where they project possible future climate and emission scenarios. So until a few months ago, we were supposedly on that SSP2, so that's called the middle of the road scenario. It's not a good pathway. We wanted to move to this side here, SSP1, to stay within 2 degrees warming. So we need to move in that direction. So this pathway is called taking the green road. You can see the keywords are sustainable development, effective international cooperation and all these things. And it seems to me over the last few months, we've taken quite a big jump in that direction here. So this is called the Rocky Road. It's characterized by policy focused on security, barriers to international trade, high inequality. So if that sounds familiar, I think we are now moving in that direction. It's not good. So most scenarios under this pathway lead to 4 degrees warming by the end of the century. The world is going to look very different then. So we've also got conservation worries around here. That's probably very familiar to many of you. So this graph here shows the populations of the white and black rhino in Kruger National Park. And you can see the populations are just collapsing, and that's due to poaching. And you might say, well, 60 years ago, there weren't any rhinos in Kruger, and that's true. So they did go extinct earlier. But what has changed is that this time the rhinos have also declined in other places around Africa. And by now, Kruger is home to a huge proportion of the global population of the rhinos. So if we lose them again, we might lose them for good. And the African penguin is another species that we might lose in our lifetimes. But I don't want to dwell too much on gloom here. So let's talk about what we can do to support conservation. So this is the only equation in my talk, and it's a simple one. So I want you to keep it in mind. So from the previous slides, you saw that often we are interested in population change. We want to find out why our populations declining or increasing, and the change in population size, delta n, is completely determined by 4 things. It's the number of individuals that are born, plus the numbers that immigrate, minus the numbers that die, minus the ones that emigrate. So if we know these 4 quantities, we know how the population is changing. And so a lot of my work is about estimating either some or all of these quantities. So keep that in mind. Then this slide here, I can actually skip. Thank you, Hussein, for covering this. But I was going to quickly give you an overview of my academic journey. Hussein has done it for me. But I'm very, very quickly going to show you a few slides of about the projects that I did before we came to Cape Town, Birgit and I. So I did my master's in Norway, as you heard, on this archipelago called Helgeland. It's about at the polar circle in the middle of Norway. And I was studying the metapopulation dynamics of house sparrows. So this was one of the field sites. I spent the summer traveling between islands there off the coast, bringing house sparrows and studying their population dynamics. It was an amazing place. Then for my PhD, I went back home to Switzerland, and Hussein also mentioned it. So I studied frogs, and I was interested in, so frogs start their life as tadpoles in aquatic ecosystems. They are usually herbivores, and then they undergo a fantastic change into becoming a frog. We call that the metamorphosis. And it completely changes their lifestyle, what they eat, where they live, what they do. And so what I did in my PhD was testing theories about how they time their transition from tadpoles to frogs. And my PhD was very experimental. So that gave me a lot of experience now to teach the experimental design part in undergraduate stuff. And you can see, including some catastrophic events here. That was one of my experiments destroyed in a winter storm. And all about randomization, blocking, replication. So you can see in those tubs, there are these little floating cages. Those host dragonfly larvae, some of them. Others are control cages. And what I was interested in is looking at how the effect of having a predator in the system affects those life history decisions. They do make decisions, maybe not consciously, but I would still call this a decision. And I was interested in so if you raise your tadpoles with a predator even if it's caged, the tadpoles will hide. They will grow more slowly, and it affects their lives in many ways. And that's what I studied for my PhD. I think this thing is a bit over. Can I take it off? It's over the microphone, I think. Good. But what I couldn't do is I couldn't really look at population dynamics. So what I was interested in is how do these predators, without even killing their prey, how do they affect population dynamics? So my next project in Canada with Brad Anholt was on a very different system. It's a ciliate. So these are protists, and they're predators here, turbularian worms. So this is all very microscopic. And you can see also my field site is shrinking here. But so these little guys here, it's not to scale actually. The worms can eat those. So that's their mouth, that they can gobble them up. But what these little unicellular organisms do, they don't just sit there and wait to be eaten. They actually, if you put them together with a worm in a drop of water, after a little while, they start looking like this. So they reshape their cell, and they make these wing-like structures. And now the predator can't eat them anymore because the predator is gape limited. So it can't swallow these guys. So I had the perfect system of looking at how these defenses can affect population dynamics and replicate it in these different vessels here. So if you're interested in watching predation, forget about Kruger. Get the microscope and a glass and a drop of water, and there's a lot going on in there. Okay, so this was a very quick overview of the stuff that I did before we came to Cape Town. And I just quickly want to talk about why do we even need stats at all? And this is a question that I usually pose to my class when we cover probability. So the question goes, imagine you're in a hospital, a heart attack patient arrives. Which of these two is more likely? A, it is a man over 55. B, it is a man over 55 with a history of heart problems. So can I have a quick show of hands? Who thinks A is more likely? Good. Who thinks B is more likely? Okay, good. Yeah, no, it's interesting. Very often I find that people choose B over A, and that's just because we tend to think in stories. So we tend to think about the story. But if you and most of you probably thought about it this way, that this group is actually a subset of this group. So every man over 55 with a heart problem is also in this group here. So there's no way that B can be more likely than A. But what this illustrates is that the way we are thinking, it serves us really well in most situations, but then our intuition fails in very specific ways. And statistics and probability gives us tools that help us in exactly those situations where our intuition fails us. So that's what makes statistics so powerful. Here's another example. So which of these sequences are random? Can you see any sequences that are not random on here? Well, actually, they are all random. So these are all random sequences of zeros and one. In those three sequences, the zeros and the ones are about equally likely. In this sequence, the zeros are more likely. Here, the ones are more likely. But these are all random sequences in the sense that each value does not depend on the previous value. And what I wanted to illustrate here is that our brains are very good at finding patterns where there are no patterns. Well, often there are patterns, but we are also finding patterns where there's just noise, where there's randomness. And again, statistics gives us the tools to separate out those situations where we are looking at random noise. Okay, so with that out of the way, let's talk about some of the research. But at this point, I need to say, so Brad Arnold, my mentor in Canada, he once said the definition of a professor is the person who's taking credit for their students' work. So I need to confess, most of my stories are about stuff that my students did. Yeah, and but I'm going to try to give credit, so hopefully it will be clear. So Brad also said, if you want to be successful as an academic, you need to surround yourself with people who are smarter than you are. And I think I did very well on that account too. So there are, behind every story, there's actually a big group of people, many collaborators. And I've made the decision not to try to list my collaborators. So those people are there, but I was afraid I would leave out too many people. So I'm not going to even try to list my collaborators. So they are acknowledged, but you won't see their names on the slide mostly. Okay, so the first story is about Rose's mountain toad. So these are small voiceless toads. You can see the size of them, and they live only on the Cape Peninsula. So we only know of 2 populations where they occur. One of them is in Silvermine, and we still need to go up and check. It's a place that, well, Ben is going to go and check whether they were affected by the fire that recently went through. So a few years ago, Francois Becker did his honors project on these frogs, and his project was about estimating survival in these frogs. So I should also mention that these frogs breathe in small puddles, and that's where their tadpoles develop. So that's going to be important to the story. So the data that we have look like that. So these are so-called capture mark recapture data. So we go out, we catch a bunch of animals, we mark them, we release them, and then later we go back and we look how many of those marked individuals have survived, how many do we find again, and we do that year after year. And then we end up with these so-called capture histories. So for example, the first capture history means these are frogs that were captured in the first year and then never seen again. So these are not random sequences now. So these are basically the data that we use to estimate survival. And we observed 117 frogs in this group, and then 7 frogs were marked in the first year, seen again in the second year, and then never seen again, and so on. So the problem now with these data is that we don't know when these frogs died. So we don't find the dead frogs. But what we do is we find capture histories like this. So these are from 12 frogs that were marked in the first year, then missed. We didn't see them in the second year, but we saw them again in the third year. So from that, we know they were always alive, but we didn't see them in the second year. And that gives us information on the detection probability, the capture probability of these frogs. So that's the probability that we catch a frog that's actually alive, which is less than 1. And that's why we get these internal zeros here. So Francois used capture mark recapture models, and then one afternoon he sent me this picture here. So he had estimated survival over the years of the study, and you can see survival goes from close to 0 here to like virtually 1. So that means in some years, almost all the frogs die. In other years, almost all survive. And when I saw this, I said, there's something wrong. You can't, I've never seen that much variation in survival before. And normally you would say, no, this species has 80% survival or whatever. But here, you can't say it goes from 0 to 1, basically, from 1 year to the next. And initially I was convinced that Francois had done something wrong. It can't be. So I spent the rest of the afternoon and late into the night, throwing every trick at this data set, trying to figure out what is going on. And then late at night, I came to the conclusion that actually that's what the data are telling us. Next morning, Francois sent me this picture here. So he said, he had realized that survival was almost perfectly related to breeding season rainfall. And again, that is for a biological system where you've got lots of variability. This is quite a remarkable result. And then the other thing is, you can see years with a lot of rain were associated with lower survival, and years with little rain were associated with high survival. So frogs like water, they need water. What's happening here? So we scratched our heads a bit. And then when Francois looked at measures of breeding effort or breeding investment, so these are all different ways. Our data were a little bit more messy about that, but in every case, there was a positive tendency between rainfall and breeding effort, breeding probability, and all of these things. And so what it turns out, what we concluded from this study was that these frogs actually have quite a unique life history. So they, in years when it's good for them to breed, so remember they breed in small puddles, so when there's a lot of rain, it's good to breed. They've got the tadpoles have got chances of metamorphosing, and that's when they invest all their energy into breeding, and then they die. A lot of them die. Whereas in years where it's not good, there's not much water, they can't really breed, they just look after themselves and they survive much better. So that's basically the conclusion. And we published that in American Naturalist. Must be one of the best published honest theses, I'm sure. So it's very chuffed about that. Okay, so from frogs to rhinos. So a few years ago, I was involved with the Rhino Impact Investment Project. So this project was about unlocking new types of funding for rhino conservation. And it was in collaboration with the Zoological Society of London and many other people. So if you're interested in the financial details, you can Google Rhino Impact Investment, and you'll find all the explanation. What concerns us for tonight is that the idea of the project was to tie funding to favorable conservation outcomes. And in particularly they had 3 performance indicators. So the biological growth rate, so that is basically just looking at the population growth rate, just looking at births and deaths. So we want more births than deaths when the population is increasing. And then they also wanted the annual growth rate. And the difference is there that we factor in movement as well. So think about that equation that I showed you a few slides earlier. So now we are looking at everything. Also addition, and in this case, in the case of rhinos, these are translocations. So the managers sometimes buy rhinos and bring them into their population, or they catch rhinos and they translocate them somewhere else. And the annual growth rate here accounts for those changes as well. And then another performance indicator was the percentage of rhino lost to poaching. And my job was to develop a statistical model that would estimate these 3 things and would be estimating this in a very robust way, because the bankers had to be convinced to invest money. This had to be quite robust. Right, so the data we have here, well, it's a bit similar to the frogs, but now, so we've got marked rhinos that we can recite. But in addition to that, we can also find the rhinos dead. And when, sorry for the gory pictures, but when the rangers find a dead rhino, they can tell it's died of natural causes or it's being poached. So rather than just only being able to find the individuals alive or dead, we are actually we are able to see them in different states. So they can be alive, so then the capture history gets an L. They can be poached, dead by being poached, gets a P, and then D is just dead for other causes. So, for example, these are just 3 example life capture histories. This individual was marked in the first year, then seen alive in the second year, not seen in the third year, and then found dead from other causes. And then once it's found dead, it's no longer part of the population. So after that, we'll never see it again. Then we've also got capture histories like this. So this is an individual who was seen live 4 times in a row. and then it disappeared. We don't know what happened to it. And then here's one that was seen alive, not seen, and then found poached. So the problem again here is that we have incomplete information, and so there's a resighting probability or re-encounter probability of live rhinos, and that's a bit similar to the frogs. But then it's also not every carcass gets found, so we need to estimate the probability of finding a dead rhino as well. And I used the so-called multi-state capture mark recapture model. So it's those 3 states that I showed you on the previous slide with the capture history. So an individual can be alive, or it can be freshly dead because it was poached, freshly dead because it died. And the transition rates here, the transition probabilities from one state to the next estimate. So this estimates poaching mortality, this estimates mortality from natural causes. And then obviously staying in this state means surviving. And then also you can see that that's technically called an absorbing state. That's just a technical trick, bookkeeping trick to make sure that those rhinos that have died earlier, that they don't interfere with our estimation on this side. So this gives me just one of the performance indicators, right? So that's the poaching mortality. I also need the population growth rate and the actual growth rate. And for that, I've got more states. So this is basically a state, a virtual state, that means not yet born. And then the transition probability from this state into the alive state, that is what we call the recruitment probability, or it's the probability of entering the population by being born, basically. And then rhinos can also enter the population through being introduced from a different population from outside, or they can leave the population by being captured and translocated out. So we tried this model on data from Tsavo West National Park in Kenya, and my collaborators were really happy, fortunately, with how this worked. So we were able to estimate the population growth rate. In that case, the population was increasing, and they were quite happy. So it matched with what they knew about the population, and they are still using this model now in this project. And so that was work with Chris Baikiwi at the then Zoological Society of London, and now with Conservation Alpha. Okay, still staying with rhinos. So I told you the Greater Kruger area is home to a large proportion of the global rhino population, and they've got a big problem with poaching. So they are spending a lot of money trying to look after their rhinos, and they do a lot they have a lot of interventions that they do to try to stop poaching. So so there are dog units, fences, security, all sorts of things, and they also dehorn rhinos. So that means they remove their horns basically to make the rhino less attractive to poaching. And so we were approached by the Greater Kruger Environmental Protection Foundation, and they had collected a lot of data on what they did, and then how many rhinos were poached in a sort of per quarter. So we looked at the relationship between those interventions, to what degree they were done, and then how that affected poaching mortality. And you can see here the only thing for which we found evidence for that it actually worked was dehorning. So those gray dots here, the confidence interval shows there's a lot of uncertainty. It overlaps 0, which means there's no effect. Whereas on this side, we do find an effect. And so this is work by Tim Kuiper, a postdoc of mine, and the way he set up this axis is that bigger means it reduces poaching. Okay, so that was a really important insight, and then we looked at the dehorning intervention in a little bit more detail. So this is what we call a regression discontinuity analysis. So we look at the time before a reserve dehorned their rhinos and then afterwards. So I should say this includes data from 12 reserves. So it's Kruger National Park and then the private nature reserves around them. So we had quite a lot of data on that. And what we found was very clearly an abrupt change right after the intervention. And the intervention was done at different times in different reserves. So, so this is quite a convincing result of actually the reduction in poaching mortality was due to the intervention. And then based on this work, the KwaZulu-Natal nature reserves also dehorned their rhinos and poaching also dropped. But there's a bit a dark cloud now that poachers have returned to Kruger and they've started targeting dehorned rhinos for, like, there are little stumps of horn left. And so, yeah, so it just shows that it's really difficult to find something that works in the long run. And I guess what I'm also taking away from this is unless we can stop the international trade and international demand, there's probably little that the managers of these reserves can do to save the rhinos. So the exciting thing is this paper was accepted in Science a little while ago. It's still in press. I can't give you the full story yet. Still under embargo, but in a few weeks, hopefully, we'll be able to distribute the reprint. Okay, then the next thing I want to talk about is it's about body size. So Bergmann was a German biologist. He lived more than 150 years ago, and he observed that among closely related species and also between populations within a species, you tend to find the bigger animals in colder environments and the smaller animals in hotter environments. And his explanation was based on physiology. So his argument was that if you're a small animal, then your surface area is relatively large compared to your size. Whereas if you are a larger animal, your surface area is relatively smaller compared to your size. So, so the larger animals would lose less heat if they live in cold areas, and conversely, animals living in hot places, because they are small, they can dissipate heat more quickly. So the question is now, we know that the temperatures are increasing around the globe. So is this going to impose selection on animals to become smaller, or animals becoming smaller due to climate change? So that was an interesting idea. Joram Jontov published a paper with this idea almost 25 years ago, and he looked at bird ringing data. So when people catch birds to put a ring on, they also weigh them. And so he found in some species, he found a trend that those birds are actually getting lighter over time. And so he said, well, yeah, it's because of climate change. But I was not very convinced, because if you think about, well, first of all, there are many other things that affect body size, obviously, otherwise we would see animals of roughly the same size in one place. But more importantly, so if you think about trends in body size happening for whatever reason, if your theory just says it should go in one direction, then you're right about half the time. So I thought that's maybe not a very strong argument. And I kind of dismissed the idea for a little while, I must admit. But then I got a chance to test the idea in a much stronger way. And this was using capture data again on mountain wagtails. And Stephen Piper collected these data. Stephen Piper was a stats professor at the University of KwaZulu-Natal and one of the early statistical ecologists here in the country. And he also was an avid bird ringer, and he ringed those and followed those wagtails over 25 years. So when I first arrived in Cape Town, I was looking for these long-term demographic data sets because I was interested in demography. And I spoke to Stephen Piper, and we started working on this. And then he unfortunately passed away. So he passed away in 2008, and we were left then doing the rest without him. But in his data set, you can see the weight of the wagtails declined over the 25 years of his study. and temperature at his study site increased. There was a bit of a blip here, but generally temperature increased. So, so the question is now, is this change in body size related to the increase in temperature or not? And the prediction was that if the mass change is an evolutionary response to temperature change, then there should be a temperature-dependent selection on body mass. So that's maybe a bit of a mouthful, but basically what we were expecting is that large birds should survive relatively better at low temperatures, and small birds should survive relatively better at high temperatures. We did the capture mark recapture analysis, and that's what we actually found. So I didn't really expect it, but we found exactly what we would have expected if the body mass change was due to selection. So you can see here the dark line. This is survival against temperature, and under the colder temperatures, a big bird, the bigger bird here with the dark line, the relationship was positive. So that means they survived. Sorry, that was the light. A small bird survived relatively better on the higher temperatures, relatively less well on the low temperatures, and then the relationship was reversed for heavy individuals. So we did find some support, and the analysis, a lot of this was done by an intern during the pro course. Okay, so with this picture, I'm taking you to Inaugural Nature Reserve, and that's where David Mapiso did his PhD. So David was one of my first PhD students, and he's an amazing birder and expert of these grassland birds. So spending a day birding in the grasslands with him is just a fantastic experience. And I didn't know much about grasslands or the grassland birds, so I learned a lot from him. But you're probably all familiar with Inaugural. So Inaugural is home to a pond storage scheme. Pond storage scheme, you've got two dams, one on top of the escarpment and one at the bottom, and then water is pumped up and down, and that serves like a giant battery. So the Inaugural pond storage scheme has saved us from some stages of load shedding a few times. But back then, when David worked there, the pond storage scheme was under construction, and it impacted those high altitude grasslands. So behind you, if you turn around, you see KwaZulu-Natal at the back. You're standing on the escarpment here. And David, so what ESCOM did was they bought a lot of land and they promised that they would manage it as a nature reserve. And David's PhD was basically about establishing the scientific basis of how this nature reserve should be managed. So the management interventions that they were considering was fire. So these grasslands burn quite a lot, but how often should you burn it? And then grazing, how many animals should you have on this? And David's insight was that fire and grazing affect grass height and density, and those are the key variables that affect the birds. So he spent a lot of time doing transects and sampling birds, and then estimating the density of different bird species. And what he found was that the different species that were important there had quite different reactions to the environment. So I'm just showing you a very small part of his results, but sitting testiculars, for example, like grass height, like high grass, so they are more in the higher grass and less in a lower grass. So this is the density of the birds, the population density. And then yellow-breasted pipit likes the short grass. So he found that the different species like different habitats, and then he worked out the system of how mosaic type of management should be able to bring out the highest diversity of grassland birds. Okay, so David, he estimated density using a method called distance sampling, but then sometimes we are only interested in whether species occupies the site or not, and for that we can use occupancy. So occupancy is the proportion of sites that are occupied by a species. So imagine you've got an area here, landscape, And some of these grid cells are occupied, so that's the orange ones, and then the gray ones are unoccupied, so the species does not occur there. So if we knew about all of this, we could calculate occupancy relatively easily by just taking the number of occupied grid cells divided by the total number of grid cells. And obviously what we would be interested in here is how occupancy changes in relation to covariates, function of covariates. So this is if we have perfect knowledge, but again, we are dealing with the imperfect detection, and what we are actually seeing here is so we don't see all the species every time, and what we actually see might be more something like this. What we want to know is this. What we see is this, because our detection probability is less than 1. And if you then do this calculation from the previous slide, what you're actually getting is the occupancy probability times detection probability. And if you're looking at gradients in dark quantity, you're never sure whether you're just looking at patterns in the detection process or patterns in what you're actually interested in. So the way to overcome this is to go and sample repeatedly. So we've got the same, so that's truth, and then repeated sort of samples of this. And then what we get is for each grid cell, we get a survey history. So this looks very similar to the capture histories that we saw earlier, but now they are survey histories tied to a particular site. So for example, if you look at that site here, the corner site, the species was detected on the first visit, then not detected on the second visit, and detected again on the third visit. Its survey history is 101. And unlike with capture market capture data, we also observe all 0 surveys. So we survey history. So we've got grid cells where the species was never detected. And obviously some of them, the reason why we never detected is because the species doesn't occur. But then in other places, we didn't detect it just because we overlooked it. And the trick of these models is to tease apart which is which, basically. So that's the goal of occupancy models. And yeah, I should just say, so what we assume here is that occupancy doesn't change during your survey. So you can see those truth is staying the same. And that's what we call the closure assumption, which is really critical. And we also assume that there are no false detections. But we can relax that closure assumption by just making it over sort of shorter time periods. And then we call that a season, which could be any time period that's biologically relevant. And then between seasons, we allow, the model allows for local extinction and colonization to happen. So we can, with these models, we can then estimate extinction and colonization. So you can see that opens a lot of exciting possibilities right there. So I'm going to show you a few examples where we use the modeling approach. And we've used it quite extensively on the Southern African Bird Atlas data. So the SABAP data are a gold mine. It's so we get about 2 million records per year. It's a citizen science project. Birders go out and they collect bird lists for grid cells. The African continent, we've divided it into 5 by 5 minute grid cells. People go to a particular grid cell, collect the bird list, and then submit it to the project. So that's a project that was started by the Animal Demography Unit. And they did the first phase between 1987 and 1992. And then SABAP 2, the second phase, started in 2007 and is still ongoing. So we've got a really long-term data set that we can look at changes in bird distributions. So the first study here that I want to show you today is one that was carried out by a postdoc of mine, Guillaume Perron. We looked at a lot of species of passerine birds and we compared the distribution between SABAP 1 and SABAP 2. So we were looking at long-term changes. And Guillaume distributed those, or grouped those 130 species into 4 groups. So at the top, he calls them close savanna specialists, but these are birds that like dense vegetation. And then at the other end of the continuum are the birds that like open, like grassland birds, they like open habitat. And you can see purple here means increase. On average, we saw increases in the birds that like vegetation in those places, and those are actually the places that have seen bush encroachment and densification of bush across the country. And then we see increases in those species that like open areas, mostly in the grasslands. So it's quite an interesting pattern that we saw there. And then Christine Broms, a PhD student, she looked at the range expansion of the common myna. So the mynas were introduced into South Africa from India, I think mostly, and they are expanding. So this is an example of where we are looking at changes from 1 year to the next within SABAP 2. So this is 2008, 9, 10, 11, 12, 13. And you can see here darker means a higher occupancy probability. And so these birds have expanded mostly into the Kruger area and also westward. So 10 years ago, we should redo the study just to see how far they've come since then. And then finally, at yet a different temporal scale is Luvuyo Kani's master's that he did a few years ago. He looked at migration. So he was able to get occupancy probabilities per month. And he looked at migratory species. So this is, as an example, the paradise flycatcher, which is a partial migrant. So that means in part of its range, it stays year round. And in other parts, it comes and leaves seasonally. So here in Cape Town, it leaves during the winter. And you can see here, each row is a year. You've got the 12 months across, starting in January 2004. And you can see red means the species, it means high occupancy probability, the species is here. And so you can see how they go when it becomes autumn, they are away in winter, and they arrive again in spring. And it goes like this every year. So we can see those seasonal patterns. And he was able to study quite a few migratory birds that way. Good. So we've got a lot of data and conservation decisions that need to be made, but the connection from the raw data to the decision is often a bit tortuous road. And it's not that straightforward. We need to extract information that's actually decision relevant. And so more recently, at SEEC, we've gotten into doing those data pipelines and basically setting up a pipeline that takes the raw data, makes sure the data is clean, and then does some statistical analysis on it to then serve decision-relevant information to decision makers. So this is the birdie pipeline for water bird data using SABAP data and CRAC. So that CRAC is water bird count. And the next slide just gives you an idea of what the platform shows. So we've got species distribution maps here. So these are occupancy probabilities. And then also at specific wetlands, we've got population trends. And then we've got species richness maps. And then you can also download reports for specific wetlands, which show you which species have declined versus increased, and so on. So we're hoping that we've been working with people from the Department of Environment, Forest and Fisheries, DEF, and other stakeholders to make sure that we can serve them decision relevant information. And Pachi Cervantes, the postdoc on the project, was really instrumental in driving this forward. Okay, so the exciting thing really in my field is that we get enormous data streams, and they are just increasing. There are new ways of collecting data. For example, you might be familiar with iNaturalist, where you a phone app where you can take pictures of stuff. And then there's an AI algorithm inside the app that tells you it's likely this or it's likely that. You upload it, and then experts confirm or identify further your picture. And then another app here is Merlin. So Merlin listens to birds and then is able to classify, just based on the sound, classify the bird species. This doesn't work yet in South Africa, but on a trip to Switzerland a bit earlier. I just put my phone outside for a few minutes, and it gave me a list of the birds that it had heard. So it's just new ways for people to collect data very quickly and easily, and that leads to enormous data streams. They've got acoustic, a lot of acoustic data. So here's an example of Guy Freiner, a postdoc he unfortunately passed away last year, but he was working on machine learning classifiers for underwater acoustic data to identify dolphins. And this picture here shows a moss frog on a microphone. So we've got these new amazing methods for collecting data. Then obviously satellite data are becoming ever more available, drones. We've got huge data streams coming in, and we've got exciting new ways of analyzing them, AI, machine learning, all of that. So it's exciting times to be a statistical ecologist. And the next step here, so those eco-informatics pipelines are at the center of ecological forecasting. So what we want to do is be able to predict what's going to happen in the near future. And Jasper is pioneering that here in South Africa. So I'm sure before long, Jasper will be standing here and giving his inaugural lecture. So basically, we can build those forecasting pipelines around the data pipelines that we've been working on, and then to take it even a step further. So we've got the observations. We can analyze the data. We can detect trends. If we also add causal modeling, then that gives us a framework for attributing change to certain drivers, and that's going to be really important because people will want to know why has this happened, who is responsible, and also what can we do to reverse declines and changes. Okay, so what kind of skills do we need for this? So when we started SEEC, a group of us about 11 years ago, we had this vision here of being a group of people with skills that range from systems knowledge, so being, for example, knowing everything about the frogs, to mathematical statistics, knowing everything about the mathematics behind our methods. And the idea was that we would be successful if we were able to collaborate effectively. So we said, well, we need to have broad enough skills so that we can overlap and have groups of people working effectively. And I think that's worked really well. And what we wanted to do is be decision support people for environmental decisions. And but the other thing that I think that I'm also really happy about is how training has worked out. And that's decisions that were made long before I joined UCT. But the double major system in the science faculty allows students to take stats and ecology at the same time. So, and I see some of you in the audience here. So you're training right here in the middle, and that's really where we need the skills. So that's a smart thing to do. And there are not that many places in the world where you can do that kind of training. So I think we've really been able to place ourselves at the global forefront, not just in terms of research, but also training. And then a few years ago, we started our quantitative biology major in the bio department. And all of this was really a lot of collaboration between departments, biology and stat. And I think we should also strengthen our collaboration with other departments like math, computer science, EGS, oceanography. So still a lot to do in that field. And then I just want to end with acknowledgements. So my mentors that were on one slide, and Hussein has mentioned them, and then my colleagues, which I've chosen not to try to list because there are so many of them. I've been incredibly fortunate to be working with a lot of smart people. So it's all of what I've shown you here is our massive collaboration efforts. And then I've also been very lucky with having very good students, groups of very good students. And then lastly, I also want to thank my family. Thank you. you. Thank you.
Thank you. Thank you and good evening. Vice Chancellor, Deputy Vice Chancellor, Professor Res Altwegg. I'd also just like to acknowledge your family, Birgit, Niels, and Nora. Thank you for joining us this evening to support Res. It's just an honor to be asked to give this vote of thanks to Professor Res Altwegg for his inaugural lecture. As I understand it, I'm expected to provide some perspective on the lecture and the lecturer. Just to say, Res, thank you very much for the wonderful lecture, and I would like to thank you for the experience of being part of this lecture. The Dean has already indicated where your journey started from ecology and going all the way to Canada and ending up in our department from 2023. So I'll skip that part and perhaps just say a few reflections on what you have shared with us this evening. Your lecture has illuminated the vital intersection of statistics and ecology. As you discussed in your lecture, your research has focused on the understanding on the effects of climate change and other pressures of population ecology and various dynamics of range of dynamics of various organisms. Your reflections on the population range dynamics of various organisms and how these are being shaped by climate change and other pressures were especially powerful. By integrating advanced statistical models with ecological insight, we have shown us how data can tell a story of species under stress and how we might respond to with informed actions. These insights are not only academically valuable, but also deeply relevant for conservation efforts and policymaking in an area of accelerating change. We're excited to learn, to hear about what lies ahead. We've already mentioned about ecological forecasting and also attributing change using causal modeling. I just want to say a few things about Res. The Dean already mentioned, and as well as the VC mentioned, that Res is a director of SEEC, which he has led for over 10 years. Res is conscious of the need and the demand for qualitative capacity building in biological sciences and has led the development of curriculum and courses in the department to meet this need. As evident from the lecture, Res is a prolific researcher with an impressive track record, of which we are proud of. These qualities make him a role model for staff in the department, and he also plays a major role or a mentorship role to young staff in a department that is short of professors. There's a few things beyond academia that I would like to mention. I hope no one gets into trouble with what I'm just about to say now. Res likes to spend his free time outdoors, climbing, hiking, camping, and looking at birds, frogs, plants, insects. Really a scientist through and through. He spends many hours contributing to citizen science data sets. He enjoys reading and baking pizza. Just to end, I look forward to your continued scholarship and leadership in our department. Your lecture tonight pertains a rich and long series of important and fundamental advances in statistical ecology. And on behalf of all of us in attendance tonight, I thank you for your lecture and for the insight it has brought to us, and wish you happiness and success in all that you do in the years to come. I thank you.
Did you know dehorning is the one statistically proven intervention protecting rhinos? But you know, of late there might be some concerns. This is how I will be starting conversations tonight. I'll be talking about how when you look at predators, you're not just looking at the Kruger, you can also look at microscopic predators. Res, thank you. Congratulations for the work that you've done over the years. Congratulations to you, to your family, to your colleagues, to your students, and the various departments that you have worked with, and to your center, and also congratulations to the faculty as well. We are very proud to see you reach this point, and once again, thank you for the contribution that you have made to UCT and to the science that you have been doing all these years. And it is so heartwarming to see also how much you have not lost the fire for the work. You're still so excited about the possibilities going forward. You are already thinking about collaborating with other departments, maths and computer science and ESG as well. But also you're thinking about how to get some of your students to also progress to reach the stage that you have reached yourself and the heights that you have reached yourself. You are thinking about how to incorporate technology in what you do and leverage the advances that we have in technology, machine learning, and all of that. It's very exciting to see that, and thank you for your passion and your enthusiasm. Thank you to all of you for being here tonight and for celebrating this evening with all of us. Once again, congratulations and thank you. Let's meet downstairs and enjoy some light snacks. Much appreciated.